Hello. We're going to go over Lab 7, which is the physiology of the digestive system. I do encourage you to have your lab manual, Syracuse, open. We're going to be focusing in on exercise 28. So let's begin. Now, the objectives are to review the physiology of digestive system and the action of enzymes in food digestion. We're going to review the differences between polysaccharides and disaccharides, as well as the presence of these carbohydrates in foods. We're going to examine the effects of pH on the activity of select enzymes. Now, I want to make this clear. Since we are not going to be doing this hands-on, what I'm going to do is give you some videos to demonstrate the very same experiments or very similar experiments. That should help reinforce the understanding of the labs that we're going to be undertaking. And then you are to complete the worksheet, which is pages 521, 522, and have it uploaded by the end of the week. Now, when we talk about digestion, we don't just talk about the organs. What do they do? How do they convert food into basic components such as fatty acids, amino acids, and monosaccharides? One part of that we're going to be reviewing today is looking at controlled tests for starch and disaccharides. Now, as we've mentioned before, enzymes act as biological catalysts. Catalysts are substances that increase the rate at which chemical reactions can occur. Enzymes are substrate specific, which means that a particular enzyme will act on one, maybe at the absolute two different types of particular reactions, and that's it. So just like a key in a lock, different keys will open different locks, but no key is completely universal. With enzymes, they're very picky. They have to have specific substrates for them to cause the chemical reaction to occur. Amylase is an enzyme found in saliva. It breaks down the polysaccharide starch, which we also call amylose. As you know, many enzymes end with the suffix ase. We also find amylase present as part of the enzymes that are secreted by the pancreas in the pancreatic juice, as it's referred to, or the enzyme solution that's a mix that empties into the duodenum to begin digestion. Starch, amylose, is a polymer of glucose molecules, basically long chains of glucose molecules held together by covalent bonds. Now, the chemical reaction to break down amylose is called hydrolysis. When starch is broken down, it is broken down to dextrins. These are basically smaller groupings of the glucose. Some of them are 2, others may be up to 3 or 5 glucoses long. Now, dextrins can be further broken down into disaccharide molecules. Maltose is a 2-glucose molecule. In other words, it's a disaccharide. And basically, it's composed of 2 glucoses. Maltase is an enzyme that breaks down maltose into free glucose molecules. Now, what's going to happen is I'm going to refer and describe the experiment here. And although you don't have the lab materials with you, I want you to view at least the first few videos that are going to be in the lab video folder, because they're going to do very similar, if not exactly the same experiment. You want to be able to identify, of course, positive and negative controls for starch and maltose. There is a solution called IKI. It's also called Lugol's iodide. It's an amber-colored solution of iodide at 1% and potassium iodide at 2%, and changes to black in the presence of starch. There is another important solution that would be used. These are indicators. And Benedict's solution is the one that we're going to be talking about. It's a clear blue liquid. Heating the solution with maltose in a water bath will change the color from green to red. Now, the color variation depends on the amount of maltose present. In lab notes, you would normally write down all of your lab data in your lab exercise pages. Why is it important to conduct a positive control and a negative control? Well, for one thing, you want to be able to see what is the actual effect if there is no maltose there, or if there is no starch there. That would be for negative controls. If you had positive controls, you are able to see clearly the effects if there is maltose present for the Benedict solution test, or if there is starch for the IKI test. It also, in a way, gives you a perspective if some of your reagent has gone bad. Now, in this iodine reaction, you can see here from left to right, you have Lugol's iodine, that's the IKI solution. You have a starch solution. Starch solution with iodide gives you a black color. The yellow-orange would be your negative. The purple-black would be your positive. Now, in the digestive system, after you have treated the starch to amylase, does that possibly mean that you could be starting to form maltose? Well, there's ways to look at this by using Benedict's solution. If you look on the far left, you see what Benedict's solution looks like. That would be the equivalent of your negative control. No reaction, same blue color, looks nice, kind of pretty. Okay, move over. Go to green-yellow. What happens is that you start seeing progressively color changes that are occurring based on the concentration of maltose present. Green-yellow, precipitate, tells you that there's traces of the reducing sugar. In this case, that's what they mean by maltose. When you get to orange, or an orange-red, that's a moderate amount. But when you get brick red, that's when you've got a large amount of reducing sugar. Now, testing for the presence of starch and disaccharides in various beverages, we wouldn't be able to do. But just to give you some thinking about this, I want you to review this. Basically, to conduct tests for the presence or absence of starch and disaccharides in various beverages, we would take solutions to test. And for example, we took cola, Coca-Cola, whichever type of cola you like, it's soda. You would also take milk. You would also take orange juice. And then you would observe the changes after adding the IKI and the Benedict solution. You carefully record all the observations in your lab activity pages. To help you here, just to give you an idea about something, most cola drinks that are not diet, okay, usually are going to have fructose. And they're made from, they would say, modified corn syrup. They take the starches, they put it with certain enzymes, and it gives it a nice sweet taste. Okay? Milk, the sugar that's in it is lactose. Okay? So you can kind of think about, well, lactose is not maltose. Wouldn't that still give a reaction? Not necessarily so. And orange juice, of course, the primary sugar there is, again, just fructose. Now, what they did in the experiments is they would basically, you didn't find any starch in it. So they took these different types of solutions and put them up to the Benedict solution test. Benedict's solution reacts in a water bath with whatever materials are present. You do get a slight positive reaction. If you notice on one side of the test tube here is the blue Benedicts, but in the next one they have Benedicts added on with milk. Same here. So you get some reaction, okay? Now the other thing is to examine the effects of pH on salivary amylase activity. IKI is added to a solution containing starch, which undergoes a chemical reaction, yielding a color change, blue to black. Okay, but what if the percentage of starch declines in the solution? Okay, that's an interesting point. So if I put the enzyme that breaks down starch, amylase, into that solution, shouldn't I eventually start seeing a reduction in the amount of blue-black? And at the same time, I would imagine that over time I would start seeing an increase in the amount of maltose in that very same setup. Now there's one point to keep in mind here, and this is sort of a refresher going back to the ANP1. Amylase is a protein, it's an enzyme, and like many proteins, they can have their activity affected by either pH or temperature. Amylase, salivary amylase, once it gets into the stomach, because the pH is so incredibly low, becomes denatured, inactive. Okay, the pH in the stomach is approximately 2.0. Body temperature doesn't have an effect on amylase, but if you put it in extreme cold, such as freezing water, that is a possibility to inhibit its activity. You wanted to explain or examine the effects of pH on amylase activity. You need to review this. Incubate the tubes at 37 degrees Celsius water bath. Why 37°C? That's body temperature. Record the time it takes for the solutions in the tubes to become colorless, as you can see here. Now, you got to remember something. Just because I said that different temperatures may affect the activity of the enzyme, some of them may actually denature it. Some of them would just slow down the activity of the enzyme. Therefore, you would still have the reaction occur. It just might take more time for the reaction to occur. Now, after we've examined the effects of pH on amylase activity, if you go to page 518, you'll see the protocol. Salivary amylase from a donor can contain pathogens. So if you were wondering, gee, when you did this in the classroom, did you just have people spit in a test tube? Sounds like an interesting idea. And to be honest with you, you go back 30, 40 years, people weren't thinking about biohazards and pathogens. And it was equally common to have someone just donate some saliva into a test tube to get the salivary amylase, just like it was equally possible in a high school or college lab exercise to prick your finger and donate some blood for a blood typing experiment. Unfortunately, we're now in 2020. And so to avoid any possibility of any type of pathogen contamination, etc., we don't do that at all. We get laboratory materials provided, etc. As you notice, people will label the tubes. That helps, cuts down on a lot of confusion. And what you have are tubes that are basically identical, except for the only change, and that is the pH of the environment. Starch IKI at pH 4, at pH 7, and at pH 10. You fill with 10 mils of the amylase solution, and you review. The second experiment, you're testing for the presence of starch and disaccharides after amylase digestion, which brings me back to what I was talking about earlier. Now you label your tubes. Starch and the disaccharide at pH 4, pH 7, and pH 10. By the way, pH 10 is pretty high up there as being alkaline. And when you add 1 ml from the amylase digestion in each of the designated tubes, for the IKI test, you add 4 drops. For the BENEDICTS, you add 10 drops of it. Swirl, heat below boiling point for 5 minutes. And as you can see from this diagram, you can see some of the results. You would also see that basically, as you get cooler, it's going to take longer for reactions to occur. In extremes of pH, such as 4 or 10, you would have either delays in chemical reactions or no reaction at all. Now, I know that this seemed less hands-on, less monitoring of this and that. Review the section. Review the videos. That will help you to understand some of these concepts. One could take you through all of the different physiological processes of enzymes breaking down all the different food groups, from proteins to lipids to carbohydrates. And usually with just this basic experiment, you can see the difference between a polysaccharide and a disaccharide and the influence of just one enzyme on them, and the way that we measure the presence of carbohydrates in foods. You also get an understanding of how the effects of pH may affect the activity of selective enzymes. In the end, review this material, review the videos, complete the worksheets on pages 521 and 522. Turn in the completed worksheet by uploading it by 5 p.m. of Friday of this week. Have a nice week.